Um,
Now,
Good afternoon. It's a real pleasure to me to welcome our speaker today, Professor Matthias Matter. Matthias is Professor of Pathology at the University of Basel and head of the Molecular Pathology Unit at the University Hospital Basel with a specialization in molecular and gastrointestinal pathology. His current research focuses on liver disease, tumor immunology, and inflammatory processes in the tissue microenvironment. His group has demonstrated a strong expertise in the application and analysis of advanced spatial, transcriptomic, and proteomic technologies to dissect the immune mechanism of liver injury, including vaccine-associated liver injury and immune-mediated liver injury caused by immune checkpoint inhibitors. It's a real pleasure for us to have him here today to present this talk called An Integrated Single-Cell Atlas of Checkpoint Inhibitor-Induced Liver Injury Links Shared Liver Tumor CD8 Positive T-cell clones to cytotoxicity and macrophage crosstalk. Thank you very much for being here today, Matthias.
Yeah, thank you, Raul. Thanks very much for the nice introduction. And thank you for the invitation. It's really a pleasure to show you some of the data we have. And as you already told, so we're working on immune pathology, mainly in the liver, and the interaction between immune cells and hepatocytes and also tumor cells. So I will just start sharing the presentation. Can you see this now?
no.
No, wait a
not
second.
yet, not yet.
Wait a second. Did I do anything? Now it should work.
Now, yes.
But it's not now it should be presentation mode.
Okay,
Is this fine? Is this fine now?
fine, perfect.
Yes, okay. So I also would like to introduce Sarp, who is working with me together. He is also listening to the talk, and he has done a lot of those analyses, which are quite bioinformatic oriented. So maybe he also will be able to answer some of the questions if I'm unable to do so. So just as a short introduction, and you know this very well, is I'm in this project working on, and that's what this presentation on is to show you some data on checkpoint inhibitor-induced liver injury. So as you all know, you have drug-induced liver injury, you have autoimmune hepatitis, and there is this group of liver diseases which are drug-induced autoimmune-like hepatitis cases, and within there is also falling this liver disease, which is becoming more and more important of checkpoint inhibitor-induced liver injury. And with the increase of the use of checkpoint inhibitors, we more and more see those cases, and me as a pathologist, I see them quite regularly in patients which are treated with immune checkpoint inhibitors. These immune-related adverse events are, as you know, quite common. So the most common organ which is affected is actually the gastrointestinal tract, and if you exclude the liver, so you have in combination therapy, so patients which are treated with anti-PD-1 and anti-CTLA, for which in at least here in Switzerland, most cases are under ipilimumab and pembrolizumab. In almost about 40% of the patients which are treated with those checkpoint inhibitors, you can see immune-related adverse events. And as is seen in this graph also, it's more common, of course, if you have a combined therapy, and it's less common if you only have a monotherapy, for example, here with anti-CTLA4 or anti-PD-1. And the liver is actually a third most frequent organ to be affected. So after gastrointestinal tract and the skin, the liver depending on the publication, is affected somewhere around 20 to 25%. But it's quite common to see patients which have elevated liver enzymes. The reason why this all works, and especially the mechanisms, they are not very well understood. And most data actually up to now comes from mouse models of chili. There are a few models which more or less can be used to understand better the disease, but they're not completely equivalent to what we see in humans, but most of the data that we have is from mouse models. We have some clinical pathological characterization, and of course we have some data also from the serum, but less from the liver at itself. But what most data so far indicate is that we have an interaction between monocytes and CD8 T cells, and it's probably mainly the CD8 T cells which turn into cytotoxic T cells, and they produce granzyme B, perforin, and interferon gamma, for example, and they then affect this the disease, which is characterized mainly by hepatocyte apoptosis and inflammation of the liver. But as I say, there is still some lacking data how actually this all is working, and what I'm going to present today here is some data how we think some of it can work. So to do this, actually, we collected some liver biopsy samples. So this is a cohort that we built, and here the cohort that we are working currently is of 35 patients, more or less female and males have the same percentage. The mean age is around 60%, and the most frequent cancer type those patients have is melanoma, which is of course evident because melanomas are quite frequently treated with immune checkpoint inhibitors, and then it goes on with several different types of tumors, and second most frequent is adenocarcinoma of the lung, so non-small cell lung cancer, and we have actually only one hepatocellular carcinoma case, and all other cases, so there's one here, HCC, and all other cases are not liver cancer types, and they come from all over different organs. About half of the patients have a combination therapy, so they get two types of immune checkpoint inhibitors, and usually, as I said, it's more frequent to have immune checkpoint inhibitor therapy related effects if you have a combination therapy. Small part or a third a third of those patients have already been under steroid treatment, but most of them only for a few days. So most of the cases that we do have, they have at the time point of liver biopsy, they have elevated liver enzymes, but they have been treated in about a third of the cases by steroids. If we look at this at the liver, there are actually a few types of different immune-related liver disease that we can actually characterize. There are actually two types of currently used classifications. One is mainly based on looking at the histology. That's what we do. So so we look at where is actually the main part of the liver damage, and there you can differentiate between mainly damage which is focusing mainly on the lobules. So the immune infiltration is mainly in the lobules and less in the portal tracts, and this is mainly called immune-related hepatitis, so mainly on the lobules. And you can also find cases where the main effect is actually on the bile ducts. So you see lymphocytes which are infiltrating the bile ducts, and this is mainly called immune-related cholangitis. And you can have a mixed form, of course. So you have immune-related cholangiohepatitis. Most data indicate that immune-related cholangitis, they respond worse to steroid therapy, and hepatitis a little bit better. And here we classify them based on ALT and ALP levels. That's another way to characterize those. You can build a ratio and then define if this is mainly an hepatitis or a cholangitis. Usually the morphology is the same, but there are a few cases that maybe on morphology more you would categorize them on cholangitis, but still you see in the liver enzymes damage also to the hepatocytes. But here the liver injury is based on the ALT and ALP level. There are classifications, of course, also how to characterize the grading of the disease, and most cases here. So this is based on the CTCAE levels. They are grade 3 and 4, and those are usually the cases which are biopsied. The ones before usually are not biopsied. We have a few cases also where they just already biopsied. That was especially at the beginning, also at lower levels. But most of them have 80% have grade 3 and 4. So they're already advanced. They have quite high liver enzymes. Most of the cases, they start to show the disease more or less after a month. So in the median of immune related damage to the liver is seen after 27 days, so more or less about after a month, but it's known that this can be quite variable. You can even get, in some cases, liver disease even after stopping the immune checkpoint inhibitor therapy. And of course, those laboratory values indicate that there's a liver disease. So this is the cohort that we are working with, and we are doing several types of analysis. And this is the next slide to show this. So we have a cohort of 35 patients. You could choose for sure different types of liver disease as control cohorts, and some studies, they do this on autoimmune hepatitis. Actually, we also have an autoimmune hepatitis cohort, but in this case, we just used normal liver cohort. So these are patients which actually have a diagnosis of a tumor in the liver, mainly, or all cases are patients that have a metastasis to the liver, and they get a normal, they give a non-tumoral biopsy from the normal liver, and the liver is completely normal. So this is our normal cohort, and it's, of course, this is quite a strong difference. So we have this Chile cohort, and we have the we have a normal cohort, and you can see quite striking difference, of course, in terms of inflammation, because in the normal cohort there's actually no or almost no inflammation. We do multi-model analysis on those liver biopsies. The first part will be on spatial proteomics, and we work with a PhenoCycler fusion platform. This is mainly based on normal antibodies that you tag with barcodes, and then afterwards you can you can overlay those images that you are taking. And we work with a panel of about 40 markers. This can also be done, for example, with mass spectrometry. So this is a similar technique to mass spectrometry. They have a little bit of a difference in the application, but more or less the data analysis is quite similar if you know this technique. We also use bulk RNA sequencing just to confirm some of it. We also did then with some of the samples bulk TCR sequencing just to identify some of T cells and check if we have some clones. And in the last part, we also show some data that we use spatial transcriptomics. We use the Xenium platform. Those that are familiar with these techniques, there are several platforms that we can or that can be used. They're quite expensive, and the Xenium platform has can actually be used to really look at single cell levels. So the granularity is quite good, so you can actually go into a single cell and analyze this cell. But I'll show you then afterwards some data, and you will better understand those techniques. So this is just to show how we worked and how is our sample preparation and how we have our experiments done. So first of all, we produced TMAs because those assays are quite expensive. So we use those different biopsies, and then we put them into a TMA. So we have about 10 to 16 samples from one patient on one slide, which we then can use. and this makes things a little bit easier. We have less batch effects, and it's also easier to analyze afterwards. We just have several samples on one slide, and this is the panel that we used for this experiment. So you can see we have some markers for hepatocytes, we have markers for myelocytes, we have markers for lymphoid size, we have markers just to characterize better those T cells. We have cell proliferation, and we also have some markers for stromal and endothelial cells. This is the advantage, of course, of this technique, is to have many different markers to characterize better to the cells, the immune cells especially. So here in pathology, usually we only work with one marker, so you just know this cell is a CD8 cell, but it's difficult for us, of course, then to characterize if this is more an exhausted a memory cell or a naive cell, and this marker help you just to characterize much better those cells. However, the disadvantage of this is that not all markers work well, so you have to pay attention if that you don't have any background, and sometimes some markers just don't work well, and you are limited still. Although in theory you could use many different antibodies, but you're still limited because some markers do not give you reliable stainings. And these are just some pictures how this all works. So this is just one, we call them tissue microarrays, so TMA, and we put those liver biopsies in those TMAs, and we have controls of tonsils, for example. We have normal liver and with chili liver, so we can analyze several different types of diseases on the same slide, and these are just some markers that we use. The next slide is just to show a little bit how we use this data then to generate results. And here on the left part, you have again a normal liver, more or less, with showing a portal region and different types of immune cells that we can characterize. And in the first step, after we acquire this data with this phenocycle, this uses we need about a day to generate all this data. You do a cell segmentation, so you characterize or you segment every cell, so you identify every cell, and then you use different algorithm. We use here the phenograph clustering. This is based on Leuven clustering, just to characterize those cells and give names to those cells. So here you see an TSNE plot, and you can then characterize different types of cells. And we just do just a general analysis of those cells, so we can then characterize different cell types. So here, for example, hepatocytes, they're mainly arginase positive, also Hepar positive. So you can see here, so with using different types of markers, we can then be quite sure that these cells here, this single cell, so after segmentation, is a hepatocyte, or it's a CD8 T cells, is a macrophage, and so on. And this helps you just to get a much broader tissue characterization if you use those different markers. And if we do just a simple analysis between normal samples, so these normal samples, and compare them to the chili samples, you can see, of course, that in normal samples, you have mainly the main cells that you have are hepatocytes. So this is light blue. But in contrast, then in chili samples, you see that there is mainly an influx of CD8 T cells. You have much more CD8 T cells. And another cell type that actually also we see that increases are myeloid macrophages. So this is this pink colored cell types. So you can do, of course, many different analyses, and this, of course, will change if you compare, for example, to autoimmune hepatitis, of course. But if you compare this just to the normal liver, you can see that mainly CD8 T cells and myeloid macrophages, they show the main differences, and these show the most pronounced difference. And actually, that's what actually confirms what is already known in the literature. So CD8 T cells and myeloid macrophages seem to play a major role in this immune checkpoint inhibitor therapy-related disease in the liver. What you can do then, although you're still to some extent limited, is you can go a little bit deeper. So you can, of course, before I just showed you that these are CD8 T cells, but you can with using those different markers, you can characterize a little bit better the lymphocytes that what we did. So we wanted to characterize a little bit better the CD8 T cells and also the myeloid cells. So we can use those markers still to characterize, are these now more cytotoxic T cells? Are they more Treg cells? Are these neutrophils, or are they NK cells, and so on? You're limited. So for the CD8 T cells, we characterize the three subtypes of CD8 T cells, and also for myeloid cells with subtype 3 myeloid cells, and these are dendritic cells, CD68 positive, CD163 high cells, and CD68 and CD106 low macrophages. So we did the same analysis after subtyping, especially myeloid cells and lymphocytes. And what we could see is that what mainly increases are CD8 cytotoxic T cells. That's actually what we would expect. So cytotoxic T cells are the main contributors to checkpoint inhibitor-related immune disease in the liver, and mixed T cells. These are uncharacterized T cells. And we also have an increase of CD68 and CD163 high and low macrophages. Very simplified, this does not reflect probably the disease, but these two can also characterize as M1 and M2 macrophages based on CD163, although most people probably would say that's too simplified, but we probably would have those two types of macrophages, which are also increased besides CD8 cytotoxic T cells. Another thing that you can do with this data is so-called cellular neighborhood analysis. Many people do this with these techniques. So you can look, are there any regions within the tissue which have a homogeneous local composition of cell types? And this is all done with bioinformatics analysis. And what we could see is actually that the cellular neighborhood 3, which is seen here at the top, which is especially characterized by quite high abundance of cytotoxic T cells, so those here, but also macrophages, that this is a cellular neighborhood which is quite prominent in Chile, and others which may be characterized by neutrophils or plasma cells are less characteristic. So this would be CN5. And if you look at a plot here where you plot this, you can see that especially CN3 shows quite a striking fold change increase, and is also quite significantly changed. So what we could see here is especially that the CN3, cellular neighborhood 3, which comprise or includes cytotoxic T cells and macrophages, seems to be quite prominent in those Chile cases. And also, if you do an analysis, which cells are interacting most in these neighborhoods, that you can see that especially cytotoxic T cells are interacting with those two types of macrophage. So this is more or less reflecting, and this is mainly now human data showing this, in contrast to murine data, that actually really cytotoxic T cells and macrophages seem to be closely related and closely interacting with each other in Chile. You can do another analysis, and this is a so-called patch analysis. You can really look at clustering of cytotoxic or of other cells, and we see the most prominent clustering of cytotoxic T cells in Chile, contrast to other cell types. And you can see here, this is one of these Chile cases that you actually see cytotoxic T cells which are clustering together. So this means more than 10 cells which are together forming a so-called cluster in those Chile case. And this is in almost all of the Chile cases the case, that you have this clustering. Of course, you don't have this in normal liver samples. And you can see that these clusters quite often are interacting with CD68, so macrophages. This is also to show here that you see in those clusters, you see much more frequent, you see this interaction between cytotoxic T cells and macrophages within these clusters than without these clusters. So this we also wanted to confirm with whole transcriptome analysis, and we only did this with a subgroup of patient samples. We did bulk RNA sequencing on normal samples and on chili samples, and what you can also see here is quite similar to what I've shown you before. So pathways which are really activated are in the chili are pathways with interferon gamma, or interferon alpha. And then if you do some analysis based on the transcriptome and looking at the cells, you can again see that based on the transcriptome that you have mainly CD8 T cells, you have a high cytotoxicity score, and you also have macrophages, some monocytes, which are increased just based on the transcriptome. Of course, this is much less precise, but more or less bulk RNA sequencing reflects what we have already seen with this proteomics analysis. So this brings me to a first summary. So the proteomics and transcriptomics data indicate that chili, that's how, I know there are different names for this disease, but mainly here we call this chili, is characterized by cytotoxic T cells and myeloid cells, mainly macrophages, CD68 positive. We see that those cytotoxic T cells, they organize with myeloid cells in spatial neighborhoods. So it seems that they are together in many cases, that they are organized together, and you can find them together. And at least the data that we have, although we cannot prove this mechanistically, is that it's quite likely that those cytotoxic T cells, they interact with myeloid cells, and that's one part of the disease. So in the second part, I will go a little bit more in detail of those cytotoxic T cells. And one thing that is actually not known is how those T cells are actually activated. It seems they probably interact with myeloid cells, but it's not known why actually they actually become cytotoxic T cells. We actually then react to liver cells. And one hypothesis is this hypothesis of shared T cells, and this has been shown in other tumor types and other organ damage in immune checkpoint inhibitor disease, but not in the liver yet. And one of the case is has been shown in pneumonitis. So patients that have also immune checkpoint inhibitor related diseases in the lung, but also in the myocardium and also in the skin. These data have shown that you have T cells which have the same specificity, meaning they are shared between the tumor and also the organ that has been affected. So they did sequencing, and they could see that the T cell receptor has the same specificity, the same sequence between the tumor and, here in this case, the lung, the heart, and the skin. And of course, this would indicate that T cells which actually are able to fight against the tumor, they may be causing also the disease in the organs far away from the tumor. It's not. This is not a proof, of course, but it could be that these cells which are attacking the tumor cells are specific to some antigens which are presented in organs, and they also react to them. And for the liver, this has not been shown yet, and we wanted to find out if actually this is also the case. So we did sequencing, and I'll show you afterwards this data. And of course, we also wanted to find out what kind of phenotype those T cells have. So in case we were to find shared T cells, we wanted to find out actually, do they have also phenotype which would indicate that they are cytotoxic. So first, what we did first is we did T cell receptor sequencing from the liver biopsy, but also from the matched tumor sample. In most cases, this was not the primary tumor, so this was the metastasis, but not to the liver. So there's actually only one case where we had we took a liver metastasis. All other cases are tumor samples which are far away from the liver, and we take the RNA from the liver, but also from the tumor, and we sequenced them. We did T cell receptor beta sequencing. We did not a combination. That could actually be some of the improvements, but we only did T cell receptor beta sequencing, and we compared afterwards those transcripts and wanted to find out, do we actually find shared T clones between the tumor and the liver? And actually, this was quite impressive. Indeed, we did. So every sample that we analyzed, on every patient that we analyzed, we found T cells with the same specificity, with the same T cell receptor between the liver and the tumor. And it also seemed that they were a little bit more expanded. So it seemed that actually within the whole cohort of T cells that we had, that actually those T cells which were shared, they actually were more likely to be higher found in the liver and also in the tumor. And this is just to show a little bit how many shared clones we actually found. So it was quite variable. So between 6 and almost 200 clones were found that were the same between the liver and the tumor. And the frequency of those clones were also quite different. So some of them were only present in 0.5% within all the T cell clones, but it go up to a 5.8% of within all the T cell clones that were shared between the tumor and the liver. And this is just to show an example. These are two patients, patient 30 and 26. We actually worked then afterwards with those samples, and you can see that these are shared clones between the tumor and the liver, and they are quite highly expressed, meaning in the tumor and also in the liver, you find this clone here quite often, or it's so-called hyperexpanded, so it's more frequent than other clones. Here is also the sequence, and we will use this sequence afterwards, so you can track those T cells, and you need the sequence, and with this sequence, actually, you can be quite sure that this is this specific T cell clone, and we use then afterwards this sequence here to track those cells. One of the first things that I actually wanted to do is to find out, are those T cells actually within the liver? Because you could just argue and say, well, you have just found those T cells in the blood, and they're just within the whole body, but they do not actually enter the liver. And we're quite sure that this is not the case. We do find, of course, T cells which we can track and which are shared within the sinusoids. But also we find quite a lot of those T cells we actually enter the liver tissue. And here this is an example of a case where we used the base scope to prove that these cells are within the liver. You can produce a base scope assay, so you can produce so-called RNA in situ hybridization probes. You can design them that these probes are actually specific for your mRNA. So you can have this, what I showed you before, this sequence, and you can have probes which actually can be used to find those T cells. And in here, you can see that some of those T cells are positive. It can be better seen here in this DAPI staining, but we do have T cells or cells actually which are in the portal region and which go into the liver. And this can also be shown in the skin here with a melanoma case. So this all is the tumor, and we can see that when you track those cells, that you can also find those shared T cells within the tumor. And if you do dual staining here with the immunohistochemistry with CD3, you can also prove actually that those T cells are, or those shared clones are CD3 positive. So this proves that we can find those shared T cells within the liver. So we wanted to know a little bit more about those T cells, and we designed actually similar to this fish assay, this Xenium experiment. This is quite a new technique, and so Xenium in situ is that technique I explained before with the spatial transcriptomics. So it actually allows you to really characterize a single cell and tell if what kind of phenotype this cell has. You can say it's more an exhausted T cell, or it's more an activated T cell. And we wanted to use this technique and combine it together with probes in order to that we have and we can track those T cells. And it's quite a new technique, and there are very few publications where actually we can that also show that you can track T cells. And so how we did this is we used an immuno-oncology panel with 380 genes. Now there are larger panels that we did the experiment about a year ago. In the meantime, they have panels which are larger, several thousand genes. But at that time, that was the panel that most people would use. And we added to this panel additional genes, so we could really well characterize hepatocytes, cholangiocytes, and endothelial cells, and so on. But also, we designed T cell probes to find the shared hyperexpander clones. We did this for 10 T cell clones. They're from 3 different patients. So that's how it works. So we know the sequence as shown before from the T cell receptor sequencing, and we designed probes which actually then can track those T cells. And that's again how we did this. So we used those liver biopsy and together with this with the matched tumor samples. This assay is very expensive. Just 2 slides costs around 25,000 euros, and you can then work with this assay or with this data for more than a year, because you have so much data from your tissue, and you can use different types of analysis. I just show you one slide how we work with this technique, and every technique has advantages and disadvantages. I think it's a great advantage, of course, that you can actually find single cells within the tissue, and I'll show you afterwards data with this technique. But sometimes you have the disadvantage that you have unspecific binding. It's not very common, but it can happen. So I just show you here a little bit to give you the feeling. So we used so any T cell. Afterwards, we show you how we did this. Or here at this stage, any cell which had more than one clone transcript that we used, and we took this in our data analysis. There were a few cells which actually had double positivity. So actually, it was a cell which had not only one T cell receptor sequence, but also 2. In the liver, this was never the case, but especially in the tumor, we found this, and we just removed those doublets, just that you know how we did the pipeline. And that's how other people do, but it's very important that you really pay attention to some of those disadvantages with those techniques. We also excluded all the cells which were not T cells. There were a few hepatocytes that also show positivity. This can be interpreted as spillover. Sometimes those lymphocytes, they are very close to hepatocytes, and we remove them. And we only use the cells which are CD3 positive, and we told that these are the target T cells only if they were CD3 positive. And also we used this afterwards and confirmed this with the PhenoCycler. So you can see here, this is just how this looks like afterwards. So you have here, this is the Xenium data. You have T cells which are in yellow, and you can tell then now these are those cells which are positive for those clonotypes. So this is just one patient. There was a patient that had 4 different types of clones, 6, 9, 10, 11. That's how we called them. And if we then afterwards do a PhenoCycler experiment, we can tell actually, yes, these are all CD3 positive. And so to be quite sure, actually, we're working with the right T cells. So what we can do then afterwards, you can do an analysis. It's similar. It is very similar then to single cell RNA sequencing data. Now this is just on tissue with spatial analysis, but you can then afterwards, using algorithms, you can then characterize those cells based on the RNA expression. You can say what kind of types of cells you have. So This is now from the liver. So as of course is clear, you have most of those cells are hepatocytes, and then you can go on and characterize different types of cells. You can have stromal cells, you can different type of T cells, and the same with the tumor. And you can tell here, for example, so these are the melanoma cells here in pink. Here is breast cancer. So these were those two types of tumor types we have as a comparison. There was a lung cancer case also, there were 3 cases. The lung cancer we were unable to characterize, probably because it was a highly infiltrated tumor, and we were not able to find the tumor cells as a specific clustering. So you can, based on those expression analysis, you can then tell what kind of cell you have in there. So each cell you can characterize. So the first thing we wanted to do is we wanted to compare the T cells between the tumor and between the liver, and just tell if they are more or less the same. And we did a subclustering of all the T cells, and when we did this characterization, we could see that we have quite a lot of cytotoxic T cells. We had 2 groups. We have the exhausted T cells, we have interferon signaling T cells, recycling T cells, naive, and then also some CD4 cells. We also have these quite well-known mite cells, which are just down here. And then you can look where are your shared clones. And this is a graph of all the shared clones. And these are the red ones are the shared clones from the liver, and the blue ones are the shared clones from the tumor. And you can see that those actually are mainly in the cytotoxic T cell cluster. So this here is mainly the red one. So this cytotoxic T cell 1, which were characterized with different markers, mainly granzymes. And the second group mainly falls into these cytotoxic T cell 2. And these are actually the main groups of cells. So the shared T cell clones in the liver and also in the tumor seem to have quite a similar phenotype, and most of them belong to cytotoxic T cells. So we call them 1 and 2 because they have different expressions, but both of them are cytotoxic. So almost around 80% of all the shared T cells in the liver and the tumor seem to be cytotoxic. And this is just to show a little bit more in detail what kind of genes are expressed. So again, here, granzyme mainly are expressed, different types, and you can see that there are mainly cytotoxic. The next thing we wanted to do is we want to look a little bit more in detail on the liver T cells, because that's where we are mainly interested. So we only looked then, we did not combine tumor and liver, only looked at the liver T cells, and this helped us also to characterize a little bit better those T cells, so to tell if they're more cytotoxic. These are tissue-resident memory cells, cycling, and so on. So we could characterize those T cells a little better, and we wanted again to look at them and see what kind of phenotype they have. And again, as expected from before, the shared T cell clones, they are mainly cytotoxic. So almost, this case here, we only had 1 group of cytotoxic T cells. So the shared T cells in the liver, they are mainly cytotoxic or interferon signaling T cells, so almost 90-95%. And if you look at the non-shared T cells, this may also be some T cells, of course, which are involved in the disease, but maybe a little bit less likely. You can see that the CD8 cells, which are not shared, they are less cytotoxic. This could indicate that those shared T cells clones, they quite often have a cytotoxic phenotype, and they might be really involved in the disease, those shared T cells, at least this data indicates. What you can do then afterwards is to look a little bit more in detail, and also to do spatial analysis. And this is just to confirm a little bit and to illustrate you a little bit more, that you understand how then afterwards you can look at those T cells. So you can then again go into the liver, and this is just to illustrate this. So this is one, this is from the liver, and this is a T cell which is positive for clone 1. It expresses granzyme H, K, and A, and so on. And then afterwards, and we also confirmed it's a phenocycler, so we confirmed that these are actually CD8 cells. But then actually you can start and look at the cells which are around and do some additional analysis. And that's what we did. So we used this data to do additional spatial analysis. We actually not completely finished, but we have a much better view now how things look like, and I'll show you afterwards the data. And just for comparison and just to confirm this data, we also used PhenoCycler, and again this base scope RISH analysis, which I showed you before, just to prove a little bit that these cells are actually have the phenotype that we think. So we used a proteomic and this RISH analysis just to do this, to confirm a little bit what we see, and we can actually see that those clones that we can characterize with this RISH analysis. So we know that this is a shared T cell clone. We can characterize these cells also with the PhenoCycler. It's just a different technique. We can say they're mainly here CD8 and granzyme B positive, and that's holds true for most of the cells, that they are CD8 and granzyme B. So we have a little bit more certainty that actually most of those shared T cells, they're actually granzyme B positive, which fits very well that they are cytotoxic. Granzyme B does not work very good with the Xenium data, and this is just for a comparison and for reassuring. So this is now this data is going now a little bit more in this interaction analysis. There are different ways to do this. This is quite an evolving field, and these are just assays that you can use, or algorithms, just to do analysis on the interaction. So this cell shot, and this just is using all this data to identify ligands and receptor pairs. So just to find out what kind of interaction probably occur in this data. And what we could see is that if you use this analysis that we have, especially the interaction between CCL5 and CCR1 and others, CCL5 and CCR5. So mainly those pathways, they really pop up as to be important as interaction. We don't know right now which are the source cells and which are the target cells, or which actually are the ones that are producing, or are the ligands which are on the receptors. And if you look at the data from this analysis, you can see that actually the shared T cell clones, they seem to be quite important. So they are cells which are actually sourcing so some of those chemokines. So we identified those shared T cell actually as number one of the source cells, and the target cells seem to be macrophages and monocytes. So it's a little bit fitting what actually we have seen before. So we see that these two cell types really seem to interact with each other. And one of those interactions that really seem to be important is CCL5, CCR1. You can see here, this is the probability. So this interaction between the shared T cell clones and macrophages seems to be quite relevant. And actually, we could also prove this then also with looking again at the Xenium data, that you can see here these T cells here. So these are the yellow ones, which are CD8 positive, and these are the clones. They're quite often surrounded by macrophages, which are CCR1 positive. So it seems really that those cells are interacting, and we can also say this, get some indication from this analysis. So you have shared T cell clones, which here in green, showing that they are quite strongly interacting with each other, show those shared CD8 positive T cell clones. So that is what we currently have as data, that this CCL5 producing T cell, most likely shared T cells, they are probably interacting with CCR1 macrophages, and this seems to be important in the disease. So just to summarize everything, on one hand, we established a workflow just to track those T cells, the shared T cells. So we started with T cell receptor sequencing. We were able then just to track those cells with the so-called base scope RNA in situ hybridization. So we could actually tell that they are within the liver, and we used again this sequence then to do spatial transcriptomics with the Xenium platform, where you can actually go into the liver and actually tell this is a shared T cell clone, and you can look at the cell. And we also combined this with the spatial proteomics, so with this PhenoCycler, and you can do an overlay of those data just to get a better overview of what is happening in the liver. And this just brings me to the conclusion. So actually, what we really can confirm is that CD8 positive cytotoxic T cells seem to be really important in Chile. We have high frequency liver tumor shared T cell clones, so clones which are shared between the liver and the tumor, and it's quite likely, or at least our data indicates that they're probably involved in the disease also because they show a cytotoxic T cell phenotype. And we think that the interaction between cytotoxic T cells and macrophages probably play a role. And what we saw is what mainly CCL5 and CCR1. I know there's data that CCR2 also plays a role. We do not really see that this is that significant. This data also comes mainly also from mouse studies. As we think CCR1 is more important than CCR2, and that's what we think is the main pathway. And the fourth point is just to show that what I've seen show before, that actually we established a workflow just to track those T cells within the liver and to characterize them better. So this work has mainly been done by SARP and by Simon. So this was also a collaboration. SARP is working in my lab, and Simon Havliger is actually also a pathologist who used to work with me here in Basel, but is currently doing his PhD at the ETH in Zurich, working with Bernd Bodenmüller. He's quite a well-known researcher working in the field of spatial proteomics. He mainly works with mass spectrometry, but does a lot of analysis of interaction, very broad, not of course not only in the liver, but in many different tumor types. And these are other collaborators which helped us to do all this analysis. And I thank you very much for your attention. Thanks a lot.
Thank you very much, Matthias. A great presentation. Thank you. A lot of data. Now the presentation is open for discussion. Let me start with two questions. One of them is, did you compare the microenvironment, the immune cell microenvironment in patients who were receiving corticosteroids with patients that were naive to corticosteroids?
This we have not done. It's something we would like to, we have to do. And also we have to do analysis, of course, of patients which are resistant to steroids. We have some patients which did not respond well. It's there, as you know, from the literature, as I said, a lot of them are cholangitis patients, and it's unknown. It's completely unknown why some patients react to steroids and others don't. But that's something we have not done yet. We just didn't have the time to do this. But we have to do this, yes, to compare treated and untreated, and also patients that respond and do not respond.
And the other question, have you correlated these clones, these immune microenvironments that you are characterizing with clinical phenotypes or severity or prognosis in this patient?
Not yet, neither. So we have a separate project where we have started to do this. It's also something that is missing. No. But we have to do this. So of course, it would be, let's say, some people, for example, there is indication, for example, that CCL5 levels in the serum could be an indication of how severe your immune-related adverse event is, not only in the liver, but also in other organs. And these are things that we want to do. Yes, we do not have, of course, for all the patients, all the data, and our cohort is still quite low, but it's certainly something that we have to work on to find out, is there any correlation with clinic? Can we use some markers just to predict if they respond and the severity? Yes, this I think is clear that we have to do this, yes.
Thank you, thank you. There is a question from Guru Aifel from Nottingham. Guru, go ahead. You are mute. We don't hear you. No. No sound.
Could you write your question in the chat, please?
So yeah, the question is if the macrophages with the T cells are resident or bone marrow. So we have the impression they are they're not copper cells and they're not from resident liver. So we have more impression that they are bone marrow derived and they have an influx. So they come into the liver. That's what our data is showing us. I know people also, there are data, it's mainly in the mouse, where they say that the copper cells also play a role. We have less impression that they are involved in our data, but this data comes mainly from the mouse.
Okay, and Daniel Tifeo is also posing a question.
Hello, Matthias. Do you hear me?
Hi, Daniel.
Hi, how are you?
Fine, thank you.
Thank you so much for this talk. It gave me a lot to think about this microenvironment, liver microenvironment, and interactions between CD8 and myeloid cells in Chile. And of course, congratulations to you and your team for the data analysis because it seems complex regarding this multimodal project. Even though I'm familiar with your work, I have several questions, but I want to focus on the last thing you just said. For me, it's really interesting that maybe the macrophages that are interacting in the liver tissue with the CD8 cells do come from the blood. They are monocyte-derived macrophages. Maybe have you thought of checking with any marker describing in literature that in fact they come from the bone marrow?
Actually, we just started to do more precise analysis on this macrophage. And just today, SARP showed me some data. I don't know, SARP, do you have additional data just that you found out today about where they're coming from? We have more impression that there are M1 macrophages. I don't know if you used any more markers to characterize them.
I mean, basically now what we are checking is that especially the CCR1 positive cells, if these are more like showing M1 like phenotype, to see if these T cells are interacting with more activated phenotype rather than the separation between bone marrow and the residency phenotype. So what we saw is that the CCR1 positive ones are mainly showing activation phenotype, producing CXCL 9, 10, 11, and some of the classicals MR marker like CD8 T 86. So we believe that the CCR1 positive cells are also the one of the most critical cells that interact with the shared T cell clones. Of course, the CCR1 itself is also an interferon signature gene, so it's kind of expected that to see the CXCL 9, 10, 11 signature. I think this is one of the main things also shown in the other immune-related adverse events, that these three chemokines are commonly secreted in the context of immune-related adverse events and dominate the picture, and they recruit further the T cells from the blood stream and contribute to disease or propagate the disease even further.
Thank you, Sarp. Yes, it's really interesting. I am happy you are considering it. From what we know from idiosyncratic DILI, or at least in animal models, as you said, the infiltration of these M1 macrophages in the liver, it's the initial step also for the resolution of the episode, because when the migrating macrophages reach the liver, they tend to have a phenotype switch towards the M2, which is more related to the resolution. And I think it could be really interesting if there is any difference in the interaction of macrophages interacting with the CD8 in the liver compared to those who interacting with the CD8 in the tumor, in case they are affecting or influencing the course of the injury. I don't know. Maybe it's just something to look for.
Yeah, thank you.
And
But do you have any other question?
the last question, sorry, because I had a lot of notes taken, but as Raul said, it was also interesting to check about the severity of the episode, because you mentioned that it's a diversity on how many shared T-cell clones you can find between patients. Maybe those with higher numbers or higher abundance of these shared T-cell clones have worse prognostic or worse course of the liver injury, because you mentioned also that they are more cytotoxic than common CD8 cells. So maybe there is something that can influence the disease severity with this parameter.
Yeah, sure. I think with this, we have to do more analysis on this. Actually, I don't think we have seen anything just with the number of T cell clones, as far as I know. SARP, no. But
Yeah.
there's something we have to do more analysis on this, yeah,
Yeah, also
to check if we see some correlation, yeah.
Also this count numbers, pure numbers based on the library size as well that you do. So if you have a maybe larger liver or the larger tumor sample, then the total TCR that you get on the shared clone might be higher. So maybe the frequency can correlate, but we haven't looked at it yet. I'm not so sure if this will show us something about the severity, because I think other cytotoxic T cells, not just the shared clones, play important role. Maybe the shared clones just initiate the disease, the others propagate the disease. So I'm not optimistic about it, actually.
Okay, understand. Thank you so
Vale.
much.
Okay, Vanessa raised her hand. Vanessa.
Hi.
And after that, I'm Daily.
Hi. Thank you, Matthias, for your excellent talk. I was wondering about the antigenic drivers behind these shared T cell clones between the liver and the tumor. Do you think they recognize like a common antigen in both tissues, in the liver, in the tumor, or could they, like this cytotoxic phenotype, reflect like a general or bystander activation in both?
Yeah, that's so. We will submit this quite soon, and this is probably quite obvious that reviewers will ask us this, and we are not that far yet to give you an answer about this. I mean, I probably would say most likely it'll be a shared antigen, shared antigen, and there's data for other organs that these are shared antigens between, for example, the heart and the tumor. But I've seen another question also a little bit related to that. So most of those, so we have a wide variety of tumors. So there's only one case with HCC, and it's known that if you have HCC on the immune checkpoint inhibitor therapy, there's a higher chance to get liver toxicity. We have a high variety of tumor types, and still it seems that we have probably some shared. I think it's most likely that we will have that these antigens are shared just between the tumor type and the liver by chance, or they are very similar. That's how they are activated. But how this actually works, we don't know. Actually, in the grant where Raul also was involved, what we plan to do is design in vitro different types of T cell receptors. You can do this with mechanistically, so you can have different types of T cells, and you can then do in vitro culture with hepatocytes, and then you can quite well know then what actually is the antigen that is driving your T cell to be activated, and that is something that we don't know. So there are experiments that you can do a T cell receptor library, so you can have all different types of antigens in more than 1,000 T cells, and then you co-culture this with hepatocytes from the same patient, and we're currently collecting samples for this just to find out better what actually is driving those T cells. And right now we don't know. Yes, of course, you can do comparison analysis. That's what is done. between, for example, RNA data. You can compare liver and tumor. It's something we have not done yet because they're all different types of tumors. But it's something we don't know, and nobody has done this, at least not in this, at least not for the liver.
Okay, I understand. Thank you.
Okay, Jan.
Okay, thanks. This is a wonderful talk, but I have a question about more classic idiosyncratic DILI. Obviously, that doesn't involve a tumor T cell response, but many forms of idiosyncratic DILI are highly associated with a T cell response, probably in most cases CD8. So, to your knowledge, has anybody done anything similar on DILI due to small molecules and looked at things like T cell clones in the liver and their interaction with other immune cells?
I think there are a few papers about clonality, but you probably, Raul or Daniel, you know this better than me. I think there are a few papers, but there's not a lot of literature about this.
Yeah, because DRESS is also fairly similar in mechanism in that we're getting T cell responses in several different tissues, including the liver. So there probably are parallels that should be looked at in the two types. And one final question is, have you considered HLA genotyping of the cases of chili? You may not see anything, but I just think for completeness, it would be interesting to know what the HLA class 1 genotypes particularly are.
Yes, yeah, you're completely right. No, we have not done this yet. That can be done quite easy.
Yes.
Mhm. Thank you. Yes.
Thanks.
There is another question coming from Guru in the chat.
I think, can you hear me now or not?
Yes.
Yeah, I don't know what I did. I probably had another Teams were on. So other question, I think I heard the answer. Matthias probably picked up the question I had written. My question was that, is it so surprising if you had a liver tumor you are treating, you had a shared clones, but it will be a little bit more interesting if you had alternative tumors. But many of them are given when they had a liver metastasis. I mean, melanoma, commonest indication for checkpoint inhibitor is that they already have a tumor in the liver, and that is what is really almost like the simplest explanation for shared clones rather than something more pathogenetically unique. So do you agree or do you have a challenge?
So I agree. I did not show the data, but most of our patients, I mean, of course, you could say they have tumor cells which have gone through the liver because they are metastasized, and it's possible that they interact within in the liver, and we just at the time point of those patients, most patients do not have a liver metastasis that we see at least by radiology. But of course, you could imagine that at least they have interacted in some ways with liver cells just by the bloodstream. That could be an explanation. I agree with you. So it's something that I think is right now unclear. But you could. It's true, of course, that almost all patients that we have, they have metastasized advanced disease. And you could imagine that, of course, they in some ways have interacted with the liver, not forming a metastasis, because we do not see that by radiology, but just have interacted with some cells of the liver. Yeah, that could be. I agree with you. That is something that we right now cannot exclude. Yeah.
Thank you.
Okay. If there is no more questions.
I think there is another question, Raul, in the chat from Yasemin Balaban.
Okay.
The question is there an explanation why autoimmunity is organ-specific, although there is shared antigen of cancer tissue?
So it's something also that I cannot answer right now. So the mechanism for the different type of organs, they probably are not the same. So I did not show you this data, but we have from quite many of those patients, we also have biopsies from the colon, because they quite often have also diarrhea, and they have also effects in the colon. And so far, we do not find, at least not this mechanism of shared T cell clones in the colon. So maybe we're not sensitive enough, and we have maybe not analyzed enough patients, but it seems that in the colon, there are other mechanisms that may explain the colitis. It seems that maybe this shared antigen is not working for all the organs. It seems to work in the same way from the heart. So these are it has been shown also for the lung. They have identified some of those antigens which are present within the tumor and the heart and the lung. And it seems that this could be a way how this works. But these questions of antigen, I think, is right now not easy to answer. And it's certainly like this, that of course certain antigens are differently expressed in different types of organs. So myosin, for example, is typical for that. Seems to be one of the antigens in the heart, and I guess myosin is very little expressed in the liver. So this probably explains why some organs are affected or not. But it probably does not explain everything. It's probably too more complex.
Okay. So I think it's time to finish the session today. Thank you very much for this great talk. Guru has another additional question. Go ahead, Guru.
No, it was just thumbs up. Thank you for
okay. Okay. So thank you very much for this great presentation, and thank you, the audience, for making possible this lively discussion, very interesting discussion. It has been a privilege, really. Thank you. Looking forward to meet you very soon.
Yes, me too. Thanks a lot. Thanks a lot
Bye.
for listening.
Bye.
Yeah.
Thank you.
Have
Thank
a nice
you.
afternoon. Bye-bye. Goodbye.
Bye.